Hello, my name is Alexa Armstrong and I'm a wetland ecologist with Colorado Natural Heritage Program. The following presentation will focus on the methods used to collect a plot-level plant species inventory. Plant species inventory and identification is considered a core method within the riparian and wetland AIM protocol. You can follow along in section 6.1 of the field protocol for lentic, riparian, and wetland systems. A plot-level vascular plant species inventory provides a rapid assessment of species richness. A thorough search of the plot can detect less frequently occurring species that may not be recorded along the transect. The timing of species inventory during a sample event is flexible depending on site conditions. In sites with lower diversity, the full species inventory should take place before starting line point intercept or LPI. In sites with high species diversity, it may be most efficient to conduct a preliminary survey of the most dominant species and to familiarize the crew with species found along the transect before starting LPI. The full species inventory protocol can then be finished after LPI. Whenever possible, plants should be identified to species in the field and recorded using codes from the U.S. Department of Agriculture Plants Database. It is important to gather the materials needed to successfully complete species inventory. Materials include the species inventory and unknown species datasheets, or an electronic device such as an iPad with appropriate Survey 123 forms for paperless data collection, stopwatch, pin flags, camera, plant identification keys and floras, a hori-hori soil knife, plastic bags, masking tape and marker, small ruler, hand lens, and cooler. For pressing plant specimens, materials include plant specimen labels, plant press, newspaper, and small envelopes. Step 1 is to complete the top section of the species inventory datasheet, including plot ID, observers, and visit date. Step 2 is to systematically and uniformly search the entire monitoring plot for vascular plant species. Focus on the area between the transects and avoid a 2-meter band on either side of the transects to prevent trampling. Work from the center of the plot toward the outer edge of the plot in a systematic or zigzag search pattern. You can see examples here of search patterns for different plot layouts. You should search for at least 30 minutes with successive 10-minute increments as needed to detect the majority of species. And after the first 10-minute increment of active searching that does not find more than 3 additional species. The plot can be searched by one person or all members of the field crew, with one specifically acting as a recorder. Step 3 is to record each species found within the monitoring plot. Species can be rooted inside the plot boundary or overhanging the plot. For example, if a willow is not rooted within the plot but is overhanging the plot boundary, the species should be recorded. Here's an example of a completed species inventory datasheet. It is important to record the scientific name and USDA plant species code if known. Unknown species encountered during the search should be flagged for later reference or collection. Step 4 is to estimate the cover class for each species found within the monitoring plot, based on its canopy cover. It's important to note that we are estimating absolute cover, not relative cover. Absolute cover is the cover of an individual species as a percentage of the total sample plot area, expressed as a percentage ranging from 0 to 100%. Absolute cover, summed across all species within a sample plot, may exceed 100% due to overlapping canopies. The cover classes are as follows. Cover class 1, present or less than 1% canopy cover. Cover class 2, occasional or 1 to less than 10% canopy cover. Cover class 3, common or 10 to less than 50% canopy cover. Cover class 4, ubiquitous or greater than or equal to 50% canopy cover. The visual estimate guide here can also be found in Appendix N of the protocol. The Survey 123 form for species inventory is pictured here. It is recommended to collect species inventory on a paper datasheet first and then transfer the data into the Survey 123 form. Species and cover classes can be recorded here. On the final page of the form, you will be provided with a summary for all plant species encountered on the monitoring plot, including unknown species. You can use the summary list to ensure all species from your paper datasheet are entered correctly into the Survey 123 form. Step 5 is to attempt to identify all unknown species with greater than 1% canopy cover and record known information. If field identification is possible, erase or cross off the temporary name and replace it with the fully spelled out species name and USDA Plants Database species code. If field identification is not possible, retain the temporary name and fill in an unofficial unknown code. You'll see a list of official unknown codes here and in Table 10 of the protocol. Unknown codes for each plot are constructed with two pieces of information. First is a set of letters to convey the level of information known about the species, such as life form, genus, or family. After the letters is a 2-digit collection number assigned to each unknown species. The number should begin at 01 for each plot and continue through all unknown species in the plot. The unknown codes for any given monitoring plot will only apply to that plot and should start at 01 for each additional monitoring plot visited. Step 6 is to collect specimens of unknown species with greater than 1% canopy cover for later identification. Before collecting, ensure that laws allow you to collect specimens. Some projects and areas have regulations that govern where and how specimens are collected. Be aware of rare plants in your region and do not collect those species. If a species with greater than 1% canopy cover cannot be identified in the field, collect a specimen from either within or surrounding the plot. If the plant species is uncommon, only collect a specimen if you observe more than 10 individuals on the plot. Collect as many features of the unknown species as possible. Roots, stems, leaves, flowers, fruits, and seeds are all valuable plant parts to collect for successful identification. Label each specimen clearly with plot ID, date, the unknown code used on the datasheet, and the name or initials of the collector. Write label information on a long piece of masking tape or flagging and wrap around the base of the specimen. Place the specimens in a sealed plastic bag and place in a cool environment out of direct sunlight. If the temperature is very hot, it is advisable to have a cooler with ice available to store plants in the field. Step 7 is to fill out a section of the unknown species datasheet or the appropriate Survey 123 form for every specimen collected. Both the unknown species datasheet and Survey 123 form are shown here. Step 8 is to take photographs of unknown plants if field collection is not possible. It is important to capture diagnostic features of the plant, such as leaves, stem, and floral parts. Capture multiple photos of plant parts and use a ruler or other object to show size and scale. Step 9 is to identify unknown species after leaving the field site. This can be done in the evening after sampling the plot or at the end of a multi-day sampling trip. If identification is possible, do not erase or cross off the unknown code on the species inventory datasheet or the Survey 123 form. Simply update the form to include the species name by selecting Final under Identification Status. It is important to include the date identified and the name of the person who identified the plant. Step 10 is to press collected plant specimens. If identification out of the field is not possible within a few days of collection, press the specimen to preserve it for later identification. Reference the protocol to read about the process of pressing a plant specimen in detail. Step 11 is to dry the pressed specimens in a warm, dry location. Ideally, full plant presses will be returned to the base location after a few field days and placed on a plant dryer that provides steady bottom heat. If a plant dryer is not available, keep the press in a warm, dry, well-ventilated location. Check the press every couple of days and replace wet blotters to speed drying. Once the specimens are dry, remove them from the press and keep individual specimens within their labeled newspapers until they are identified. Here you will see quality assurance steps you can take to ensure quality data collection in the field. These steps are also listed in the protocol at the end of section 6.1. It is recommended to reference these steps during and after each sample event. This concludes the methods for plant species inventory and identification. Thank you for listening.